Hey guys, this is our next video in the Biology 30 series. Today we're going to start talking about sex linkage and sex-linked traits in genetics. And so sex linkage is really just that. It's a trait that is linked on a specific sex, um, and not on both necessarily. So it might be passed on only in females, it might be passed on only in males. I do want to just iterate then that in terms of genetics and sex linkage and sex linked traits, we are specifically discussing uh, like gender or biological sex rather, in that binary. So in genetics, it's either genetically a male or genetically a female, and that's to say you are either a double X if you're a female or a X and a Y chromosome if you're a male. Um, so we want to keep that terminology as specific to the biology and the genetics as possible. Um, and so as we move forward here, chromosomal theory is a theory that basically dictates that, hey, chromosomes carry genes, which are units of heredity. And we know that, right? Information is carried on those genes. And you can pass those genes on to your offspring, which means that those traits are heritable. Homologous chromosomes segregate. So during meiosis, the process of going from 2N to haploid or 1N, uh, homologous chromosomes are going to randomly separate. They segregate individually. And these gametes are going to carry half the number of chromosomes as their somatic cell or body cell counterparts, right? So a gamete is haploid and a somatic cell is diploid. And so breakthrough work was done by Thomas Hunt Morgan. And um, Morgan was a guy that, kind of like Mendel, really liked to work with, well, in this case, flies. And so in 1908, Morgan worked on fruit flies and realized that, well, you know what? Certain traits are very deeply set in genetics and more specifically sex based genetics. And so we picked fruit flies for a number of reasons. First of all, they have a rapid life cycle. They live very short lives, between 10 and 15 days. This is helpful when you're trying to go through multiple cycles of generations to see how a trait is passed on. By having a lifespan of 10, 15 days, you can really see how a trait is passed on, what it looks like in that offspring, and then you can see 15 more generations, for example, very quickly. They lay hundreds of eggs at a time, so you have a lot of offspring produced. You really get to see the probability in what traits are expressed for a large number of offspring. You can really do cross experiments, so you can mate one genotype phenotype of a fly with another genotype phenotype that you want of a fly, very realistically, given that there's so many of these flies produced at one time, and you can really repeat these experiments. They mature quickly. The males and the females are significantly different in traits, much like in humans, which makes this a great way to study how traits are passed on um, genetically through sex linkage. So there's a difference between male and female fruit flies. The specifics don't matter for our course, but just know that those differences are what make these flies great among all these other reasons here. They're small, so you can store thousands of them, and they don't take a lot of room. And they have quite significant contrasting traits. We'll focus on eye color significantly here, and the eye color can vary immensely. They have a small genome, only 8 chromosomes, so 4 pairs of chromosomes each. So Morgan discovered a couple things. Primarily, the females had those 4 homologous pairs of chromosomes where, of course, 3 were autosomal, So three of their four, of the female's four, were autosomal or body cells, body genes, and one last pair of those four was the sex pair. So in this case, XX. Males only had three pairs. They had three autosomal pairs, and interestingly, like in humans, the sex chromosomes were not homologous. So in other words, they were uh, one of each. So we had one X and one Y chromosome, just like in humans. And so, like I said, a common example here is eye color in Drosophila, which is the fancy word name for fruit flies. And so let's let big R code for a red eye color. Little r is the recessive trait for white eye color. White eyes are mostly found in males, and we find that the red eyes are mostly found in the females. Now, the question is why? Why is there a sex link between which traits phenotypically for eye color showed up more commonly in which sex? Why were males more commonly having white eyes and females more commonly having those red eyes? Well, this is what sex linkage is all about. So assume these alleles are found on the X chromosome only. So let's assume for a second that the red versus white eye color is on the X chromosome only, not on the Y. If we look at the two crosses, you could cross a homozygous dominant red female, right? That's going to be this female here, XX, and you're going to cross it with a male, XY, who has the white eye allele. Notice how we're writing a superscript above the X chromosomes specifically, or the X alleles specifically. And the reason why we're writing that little superscript there on top of the X is because we're assuming that this allele is going to be found on the X chromosome only in this example. Another cross we'll look at is going to be, what if I took a heterozygous female, XX, but she's got the red allele and also the white allele, and that's crossed with a male fruit fly, XY, who now has the red eye allele on his X chromosome. So let's do that first cross. I'm going to cross the white-eyed male on the left here with the red-eyed female, and we're going to make that cross. Top down, left in, top down, left in, top down, left in, top down, left in. When you do your Punnett square here, you have 4 possible offspring. Let's investigate them. Well, first, for sex, 2 of them are female, right? XX and XX. These 2 are female. And these bottom 2 have XY, X and Y. So they are male offspring. So if we were to reduce our ratio, we not only have 50% female, 50% male, but these 2 females are exactly the same phenotype. They both are heterozygous genotypes for the red phenotype. They have red eyes. Same with these 2. These 2 are males, but they both have the red eye allele on their X chromosome. Therefore, these 2 males have red eyes. If we were to cross another generation of those 2 offspring, we would see a slightly different cross. So I still have 2 females and 2 males, but now their phenotypes look a little different. I have 2 red-eyed females. Both these females on the top here have red eyes. This one is heterozygous for the red eye allele. This one is homozygous for the red eye allele. This one is purebred. This one is going to be a carrier for the white eye color, but it won't show it. It's going to show the dominant red eye color. These 2 male flies, this one is going to have a red eye color. This fruit fly is a male that has the white eye color. So Morgan concluded then that genes controlling eye color for Drosophila, or fruit flies, must be on the X chromosome. The Y did not carry the gene for eye color, so the male's Y chromosome never carried a specific allele or gene for eye color. Males can't, by definition, be heterozygous for an X-linked trait then. Because if it's X-linked, the only way you can be heterozygous is if you have two X's. Males don't have two X's. In fruit flies or in humans, we have one X and one Y as a male. And so if we look at this chart here, females have three options. They can either be homozygous dominant, X big R, X big R. They could be heterozygous, but phenotypically show the dominant trait, so big R and then a little r. They are a carrier for that recessive trait, but they don't show it. Or the female can be recessive, homozygous recessive for that recessive trait. So if you notice here, a female is going to have a 1 in 3 chance or a 33% chance of showing the homozygous recessive trait if it's X-linked. The males, on the other hand, only have two options. Since males are XY, the male is either going to have a capital R on his X chromosome or he's going to have a lowercase recessive r on his X chromosome. He either has red eyes or is affected by the trait or not affected or has the recessive trait. So in this case, you either have a red-eyed male fruit fly or a white-eyed male fruit fly. You can never have a carrier male fly. So that's for X-linked chromosomes. Traits that are controlled by the Y chromosome are therefore only going to ever affect a man. Male flies or humans, if it's a trait that's passed on the Y chromosome, only males have Ys, and so only they can be affected. A female will never be affected with a Y chromosome. So sex-linked traits, we in humans have chromosomes 1 through 22, which are autosomes, and then the sex chromosomes are that last pair, that 23rd pair. Sex-linked traits are controlled by genes on the sex chromosomes. Humans have 22 pairs of autosomes and 1 pair of sex chromosomes. Males, as we've said, just like in Drosophila, are X and Y. Females are XX. Sex-linked traits are almost always going to be on the X chromosome. Very rarely will we have a Y-linked trait. Doesn't mean it's impossible to have a Y-linked trait, one that only affects males, but it's very rare. Most sex-linked traits are going to be on your X chromosome. The Y chromosome, therefore, and we'll discuss this in significance in our reproduction unit, but the Y chromosome is usually specifically for sex determination. So if the Y chromosome is present, it contains traits and genes that code for traits that are specifically going to help the organism develop into male-like traits, male-like phenotypic traits. And so sex-linked traits, therefore, are going to occur more commonly in males than in females because males either have it or they don't. They have a 50% chance of showing an X-linked trait versus females have a 1 in 3% chance of having the trait. So let's do a quick question here. What if I crossed a red-eyed female, so a heterozygous red-eyed female, I have to be specific here, and a white-eyed male? Well, my white-eyed male is easy, X and a Y, and it's white-eyed, therefore this fly is going to have the little r attached to its X chromosome. The red-eyed female is heterozygous, so it's going to have XX, 1 dominant r, and 1 lowercase r. We're going to fill out our Punnett square as usual, top down, left in. And I have my phenotypic and genotypic ratios. Now, if you'll notice, I've got still 2 females, 2 males. However, their phenotypes vary quite a bit. This female is red-eyed, even though it's heterozygous, because it's got the dominant R, which will be expressed. This female is white-eyed because there's no dominant R. It's homozygous recessive for the little r white-eyed gene. So we've got a white-eyed female, a red-eyed female, we've got a red-eyed male, and we've got a white-eyed male. Sex linkage in humans, probably one of the more common examples, is colorblindness. genes for visual pigments responsible for perception of specifically the red and green colors are found on the X chromosome. And so I've got a little test here. If you take a look inside this circle, if you can see the number 5 inside of there, you are not colorblind. If you can't see the number 5 there, you may be colorblind. There's other tests you can do, and I will link a video to colorblindness and some helpful links in the video description below. But if we look at colorblindness, since it's carried on the X gene, we can compare how it affects males and females differently. Males are going to have, they're either going to be colorblind or they won't be colorblind. So you either are normal eye colored or you are not a normal color vision individual as a male. You can't be a carrier as a male for color blindness because you can never have 2 X chromosomes in your genotype because as a male you have 1 X and 1 Y. But as a female, you have 3 types of genotypes. You can either be a normal female, meaning that you show the capital N normal color vision genotype on both alleles. You could be colorblind, meaning that you are homozygous recessive, little n, for both your X chromosomes. Therefore, you will phenotypically be colorblind. Or, this middle one here, you could be a carrier for colorblindness. So you still have a capital N, so you do not have colorblindness. You are a normal vision individual, female. But you have a allele that you're carrying for the recessive colorblindness trait. So this carrier female would have normal vision, but they may, depending on who they mate with, pass on that trait to their offspring, and their offspring could be colorblind. So this is how you can have offspring that are colorblind while both parents are not colorblind. So hemophilia, another good example. This is when you have trouble clotting your blood. So there's a blood factor that is critical for blood clotting, and the factor is created from genes on the X chromosome. Hemophilia, the inability to clot your blood, is going to occur more oftenly in boys than in girls for the reasons we've already discussed. As a boy, you have a 1 in 2 chance, or a 50% chance of having the X-linked trait or not. But in girls, you have a 1 in 3 chance, or only a 33% chance of having that X-linked trait. So X-linked traits like colorblindness or hemophilia are going to be more common in boys or men than in women. So these would be the examples of the genotypes involved in hemophilia. Girls can either have hemophilia, not have hemophilia, or be carriers of hemophilia. And remember, a carrier won't actually have the recessive phenotype trait. They'll have the dominant trait. They'll show the dominant trait, but they're carriers of the recessive trait. So they could pass it on to offspring depending on who they mate with. Boys have only 2 options. They're either affected or they're not affected. Just to close here, I want to discuss 2 other terms. Sex-limited versus sex-influenced. Sex-limited traits are traits that are only expressed in one sex. Great example is bulls. Bulls, so male cows, they carry the gene for milk production, but they never make milk. So the only way a cow is going to express the gene, actually express, phenotypically express and code for, through transcription and translation, the products of a gene, in this case for cows, is if they're female. Male bulls carry the gene, but they don't make the milk. They don't express the gene. They don't transcribe or translate that information into an actual protein. Sex-influenced traits are more common in one sex than another. So the sex is going to influence this. Baldness is a great example. Men are more likely to go bald than women. This isn't because men have a gene for baldness. This is because men have the genes for testosterone production. Since men make more testosterone than women, men are going to have a significantly higher chance of having baldness because testosterone affects hair follicle growth over time in a man's life. Therefore, you're going to see it more in men than in women. Now, testosterone production is recessive in females. Women make testosterone, but in very small amounts. This is why baldness is linked to or influenced by the sex of the individual in this case, due to testosterone production, which is more common in men than in women. That's going to end our video for today. The next video is going to be on gene linkage. Please make sure you do subscribe and like this video so it shows up in your news feed and you get notifications when I post the next video. Thanks, guys.